Good afternoon, everyone. Okay, I still see people joining in, so let's take a moment to settle down. All right, so let's get started. So hello, everyone, and welcome to today's webinar. My name is Sanchita Bhattacharya. I'm the Scientific Outreach Program Lead for ImmPort. It's a pleasure to have you all join us for this webinar series. I'll be moderating this session. So before we go to the main presentation, I would like to cover a few housekeeping items. The presentation time is approximately 45 minutes, followed by 10 minutes for Q&A. Throughout the session, we encourage you to ask questions using the Q&A feature, and we'll try to take the question towards the end of the presentation, and we'll take these questions live. This session is being recorded, and a copy will be made available for reference later. So today we have with us the ImmuneSpace team who will present how the ImmuneSpace resource is built, highlighting the underlying infrastructure, data standards, and workflows that supports it. The team will also discuss how the data from ImmPort is validated, curated, and enhanced within ImmuneSpace and demonstrate the community-specific query and reporting features that makes this platform a powerful tool. So with that, I'll turn it over to Dr. Bjorn Peters with a quick introduction. Dr. Peter manages the HIPC Coordinating Center. Dr. Peter works on a broad set of projects at the interface of computation and experimental biology. These projects include building custom analysis pipelines for epigenomic, transcriptomic data, establishing in-house databases to integrate and analyze laboratory and clinical data. The major component of Dr. Peters' work has also been leading the bioinformatics design of the immune epitope database, many of you know by the name IEDB, which has led to more expressive control vocabularies to convey immunological knowledge, increase consistencies in data capture, and enhance ability to query and discover in the IEDB. For the ImmuneSpace project, he's working to enhance the quality and the value of data which is exchanged by the HIPC centers, while also promoting cross-HIPC collaborations. With that, I would like to welcome the ImmuneSpace team and Dr. Peters.
Thank you so much, Sanchita.
Okay, let me stop sharing.
So, yeah, it's my pleasure here to be invited to talk about our efforts that have been done in close coordination with the ImmPort team. So I'm going to talk about this ImmuneSpace project, which has been built on the ImmPort infrastructure. And so you're going to hear from me quite briefly in just a very, very broad overview. And then I'm going to pass it on. So me being the one of the two PIs of the ImmuneSpace project, along with Steve Kleinstein. And then we're going to pass it on to Brandon Ha, who's been working on much of the implementation of the key functionalities of the ImmuneSpace website and underlying database and these processes. And then Kirsten is going to talk about the process of how we get data into ImmuneSpace, add information to it, more curated, validated, and then have it in ImmuneSpace. And then Jason is going to talk about the overall infrastructure, actually. So we're going to start kind of like with what the user sees. And at the end, we're going to go into like the underlying technological infrastructure that enables all of this. Next slide, please. So the presentation today is structured into is primarily going through this use case. So this is a somewhat unusual presentation. Like, we are really wanting to show what we have done in order to tell others how, if they want to have something similar, how they could do this themselves. If you're involved in another project or so, how to potentially build on the existing ImmPort infrastructure and add something customized to a community, because that's what we were trying to do. ImmPort is set up and funded to provide like long-term storage of all the data generated by NIH projects, but given the vast diversity of data in there, it might not fulfill the requirements of your specific user community that might want to have very specific queries available on that data. So that's what we try to build on top of the infrastructure of ImmPort, using additional validation and curation, as you'll hear throughout the talks. So we're going to do a live demo of what that means, what we can do in ImmuneSpace. We're going to demo the curation validation processes, and as I said, like technical implementation will be last. Obviously, that's all built on this. Next slide, please. So starting, this is going to be my last part. I just want to introduce HIPC itself. So HIPC is a set of centers, the Human Immunology Project Consortium. I always get this wrong. I apologize. Launched in 2010, it's gone through multiple cycles, was renewed in 2015, renewed in 2022. The current centers that are funded you see here. So this has been always a set of centers that are really at the forefront of human immunology profiling. And the goal of this network is that while each of the centers performs work that is really stands by itself, by integrating the data generated by the different centers, we want to have added value. Next slide. So the goals are, one of the things is that each center studies human immunological problems where you have a perturbation, such as vaccination or infection or transplantation or whatever, and you see by profiling a number of human subjects that undergo this perturbation, what kind of things are perturbed and how do those relate to outcomes. So what we want to do is have a resource where we can compare all these different findings and then cross-compare cross potentially related perturbations by creating a centralized resource and being able to compare these things side by side. So as I said, we are studying perturbations. I mean, vaccination is the primary thing. It used to be in the first round, it was all influenza vaccination that has spread out. And at this point, there is data on things like pregnancy, which is very much an immune perturbation, even though you might not think of it as that in a primary form. So next slide. We have here an overview of what is currently in ImmPort. So the centers deposit the data into ImmPort first. This is the overall what you have, and when you look at the overall data in ImmPort, we do make up obviously it's a fraction, but a significant fraction of the data that is in ImmPort. Next slide. And this is kind of the way the ImmPort overall, sorry, the ImmuneSpace overall, or the HIPC centers are structured. They submit their data into essentially the HIPC Coordinating Center. This is pretty much our team here, where the data is then further curated and integrated into a knowledge base that is then accessible to the scientific community and can query it. Next slide. So that is where you can access it, immunespace.org. The data ends up on this website, and you're going to hear about all these steps in more detail. So apologies for being quite loose in what I'm describing, but ultimately the goal is that we can enforce higher level data standards on the data that comes in by the HIPC centers, all of which actually were mandated as they were funded to have a data coordinating center and that is going to be able to do data updating validation and standard adherence, that in the standard R01 grant might actually be a problem. Obviously, with the goal to then have these data sets more interoperable between centers. Next slide, and with that, I pass it on to Brandon, who's going to talk about how this looks like now, what we actually enable with this work for the user. Brandon, please take it away.
Great, thanks, Bjorn. So let me share my screen. Can everyone see okay?
Yes.
Awesome. Hello, I'm Brendan, and I've been working with Anais, who has done some fantastic development on the ImmuneSpace website. Today, I'll be running through a demo of the website, and we won't have time to go over every feature of the site in depth, but we'll try to highlight the data access, the new tools that build on top of ImmPort and HIPC curation efforts. So let me go ahead and load the web page. So this is the ImmuneSpace website. It houses information about the project, including different data standards, highlights the research from the different contributing centers, and hosts some tools for data access. We do have a live community-driven forum under the support link, so if anyone's visiting it and has any questions or feedback after the webinar, feel free to submit something there. So our main focus today will be the data access. That's going to be listed here under the nav bar. And I just wanted to go over a couple motivations to what's driving this. So we wanted to provide direct data access through ontology-based queries. An example of this would be like a researcher interested in bacterial diseases and would like to filter for a certain subset of participants. And so in this query in itself, bacterial diseases is pretty loose. We're taking that concept, running it through a hierarchical query so that we can resolve it down to specific nodes like tuberculosis or pertussis. The second motivation, we wanted to provide additional curated data alongside publicly available data. So that's data from ImmPort along with what we're calling signatures or the different gene sets. So these are filtered alongside with the rest of the data to allow for ease of comparison and viewing of the different conditions. And the third one we wanted to highlight today was providing a better view of the experimental design. So for this case, we're going to go over a newer graphical interface that aims to help researchers visualize the whole experiment for the different studies and kind of view what data is available for there. So with that, we'll go on over to the website to take a look at how this kind of works. Cool. So again, to access this page, it's under the nav bar, under data. We're providing a bunch of different filters. So these might seem familiar from ImmPort, but we're adding a couple other things here. So feel free to take a look at it on your own time. We can view the breadth of the data right now. So the unfiltered data set, we've broken down the data into 6 different tabs, 6 different tables. Users with a more study-centered approach can stay on the study tab, and what's available here, if someone comes in with a specific PubMed ID for a study, if they know the specific study ID from ImmPort, you can query that here as well as publication dates, authors, et cetera, et cetera. We also have things broken down into the arms, so these are the different cohorts. We currently don't have too many query functions on this just yet. We're still working on standardizing some of the descriptions and names, but for people who are interested in specific ARM IDs, they can query for that. Similarly, for participants and events, these are data that we're retrieving from ImmPort. Participants should line up with the subjects for users who are more familiar with ImmPort. And here we have the assays tab. So this, we aim to provide the data directly with the queries. So we can see here we have over 5 million data points right now, and this is an unfiltered query. It can get a little bit unwieldy without any queries. So let's just go back and apply a query just to make sense of the data. So in our previous motivation, we talked about a researcher interested in something like bacterial diseases. So we can browse things through a tree structure and select groups at a time. And this is the data that's available under this query. So we can see there's two studies, one that's looking at blood tissue studies of tick-borne diseases and one that's looking at tuberculosis. As we thumb through these, we can see which groups, which different arms are available, the different participants available in both of these studies, as well as the different events. And we can see what kind of samples were collected at this time. So we can see there's some blood serum. These are taken from Lyme disease and tuberculosis. So just a query from bacterial diseases can resolve to larger subsets of data. As for the assays, we have a lot of ELISpot data um, that's associated with these studies. For this particular query, we don't have any signatures, so we can go back and search for something that might have a signature. So in one of our examples, we mentioned dengue disease. And we have a autocomplete here that helps expand the tree. This is a more curated ontology, so hopefully anyone using this is able to browse how they got here from the tree. So as we load the data, we can see that we have 4 signatures for this particular query. And this is the new data that's being introduced to ImmuneSpace that's unique to ImmuneSpace and not in ImmPort. And this is all manually curated. We'll talk a little bit more about the curation process and how this gets validated and into the database a little bit later. But for now, we're just showing how this is presented. So where is this data coming from? These signatures have a unique ID. We can look, for example, here, like this is coming from study 1995. It's going back to the studies. We do provide individual study pages that houses more metadata and information for these particular studies. So going into the study page, again, there's more metadata. You can see the different participants, arms, events, and assays. This corresponds to the tabs from the results if you query directly on this study. This is the timeline for the experimental design. We'll come back and elaborate on this a little bit more. For now, I just wanted to talk about where the signatures are coming from, and we can take a quick example from the actual publication for this study. We go to the free text. And just to make quick sense of it, we can look at like a figure. And we're seeing different gene sets here that are also captured in the query for 1995. Great. So let's go back to the study page. So this is another new feature that's unique to ImmuneSpace. We're aiming to curate the design, experimental design, and present it in a way that makes sense to the to researchers, and also helps provide insight into what data is available at different time points of the experiment. So for this particular study, we can see there's 2 different arms. And it's a relatively straightforward study. We have occurrence of infectious disease, goes to hospitalization, study enrollment, and the different sample collections. And then the arms track if there's symptoms or recovery. And the different tools available here, we can select different tracks, and this helps highlight how this experiment progresses. Again, this is a relatively straightforward study. We can go to one that has some more added complexity. to see how this might help. So let's go back to our query. If we take a look at the arms immediately, just from the results here, we can see study 888 has many more arms. So that's probably a more complex study. So let's go ahead and filter for this. And having done so, we can see what data is available already. We have the different events, so samples collected in Sri Lanka, the different participants, the different arms of this experiment, as well as the assays. Each one of these tabs has a download available for it. So users who want to perform more analysis offline can download this data. We provide it as a CSV, exactly as the columns we see on the site. So going back to the study, we wanted to take a quick look at the timelines. This might take a second to load, so we'll just let it go. Great. And we can see here the study is a lot more complex. Again, we have these different ways of viewing this experiment. The combined graph will show all the arms in one view. You can isolate them by clicking through these tabs, and that just provides a cleaner view of the experiment. Or scrolling down, you can view the specific arm names in the description and select them to kind of group them together. So in this case, we're comparing a lot of pre-study enrollment things and how they move through the experiment. Again, the different nodes, if there's data associated with it, we have them labeled by the planned visit IDs. So these IDs should be a little bit familiar with those familiar with ImmPort data, but we're adding different linkages here. Great, so I think that just about concludes what we wanted to demo for the site. We wanted to talk a little bit more about the signatures and how that's presented and curated. So I will hand that over to Kirsten to talk about it. Thank you.
Hi, thank you, Brandon. So one of the parts that are manually curated are the signatures that usually represent study-reported findings. It's most often either protein levels or RNA transcript levels, and they are often part of figures, but only explicitly stated in the text. And so we have curators that extract those and make them available in ImmuneSpace to be connected to the rest of the data. Can I get the next slide? Thank you. So there are a number of different curation and validation steps in ImmuneSpace and along our data flow. Next slide, please. Thank you. So as stated before, ImmPort is the primary repository for HIPC data. So the first step for our center researchers is to submit their data to ImmPort, and we require them to make use of the submission templates. So they are not allowed to just attach their data. They need to use the submission templates for everything. And then they also use the validation tool that is available on the ImmPort website to just make sure that there are no obvious errors in any of their templates. Then the data is first held in the private workspace on ImmuneSpace, and once everything is finished and ready, they will let ImmPort know that the data is ready for release. And then ImmPort will run their own internal validation and standardization processes on the data. For example, that's the step where the preferred terms columns are added. And then once the data is public, ImmuneSpace will grab the new data release and integrate it into our database. And at that point, ImmuneSpace also runs their own tests. So we test for both internal consistency and also adherence to our HIPC data standards and the community ontology terms that we use. And this whole pipeline can potentially take quite a while, so that if at this very last step we discover something that is missing or that is maybe not consistent, it's sometimes hard to track the data down. So one thing that ImmPort has implemented for us, next slide, please, is an early access option. So our researchers have the opportunity to share their data with ImmuneSpace before it becomes public. And that allows us to run our validations right away and give them early feedback. And then either everything is fine, or we can ask them to make changes and to resubmit the data. So this helps to provide feedback to submitters early, but also it avoids patches to the data set after it is made public. So next slide, please. And this is how it looks like. On the study details page, you will find kind of on the right-hand side, the option to download your parsed data from your study. And then the HIPC researchers have an additional button that they can use. So when they click that, a copy of their parsed data is sent to the HIPC Coordinating Center, so to us, and then we can run the validations on that data. And we cannot just go into their workspace and grab the data ourselves. So they have to send it. They have to send it to us. But it's a great help that we have this option. Next slide, please. Right. And so we take the data that comes in from the different ImmPort tables and we combine it, restructure, and transform it as needed for our data analysis and for the visualization that you have seen on the ImmuneSpace portal. But we also add manually curated components. And we retrieve datasets components that were submitted to third-party repositories and reintegrate them. And in most cases, that is data that's been submitted to GEO. Next slide, please. Two of the components that are manually curated as part of the study information is the timelines and the signatures that you've just heard about. And the source for that information is usually the free text information that is part of the ImmPort study and any publication that is associated with that study where people might have gone into a little bit more detail. And for the timeline, we aim to extract all events that are relevant to the study and to the study timeline. Those can be sampling events, but also non-sampling events. For example, things that need to have taken place for a certain participant to be included in a study. So things that happened before, but also events like vaccination events that happen throughout the study that are not necessarily connected to a sampling event. And also the signatures, as you've seen before. So we have curators that extract the study reported findings that are sometimes only explicitly mentioned in the text and make them available and searchable on ImmuneSpace. And for information on how we retrieve and reintegrate data from GEO, I am handing over to Jason.
Thanks, Kirsten. Right, so yeah, I will tell you a little bit about how we make some of those integrations, specifically the integration to GEO. So if you're not familiar with GEO, it's a public database of expression data. And what I'm showing you on the left here is a table that already exists in ImmPort, where they go ahead and link the different experiment samples to the corresponding geo identifiers. So these are geo sample identifiers. You can then go ahead and look those up in geo, and you'll see a page that looks like this on the right that kind of summarizes that sample. Now, it's great. We have access from the ImmuneSpace sample over to the geo sample, but that doesn't get us to the expression values. So to do that, we need to take this a couple of steps further. Next slide, please. So every geosample, I think every geosample, most geosamples are linked to a series. So this is the geosample ID, and if you kind of scroll down the page, you'll see the different series that it might be linked to. And a geosample can be linked to one or more series. A series is just a collection of related samples that are generally part of one experiment. And if we go to the series page on the right here, this is what the series record looks like. And we can see it contains 30 samples, one of which is the same as that sample on the left. And then those series also contain a bunch of supplemental files that you can see at the bottom. And it's in those supplemental files where the expression data are generally stored. Next slide. Thank you. So again, we have our geo series on the left and the supplemental files down at the bottom. If we go ahead and open up one of the supplemental files, this is what it looks like on the right side. They are usually matrix files with one row per gene and one column per sample. And so it seems like it should be a trivial thing to match up the samples that are in geo to the actual columns in the matrix, but it's not always straightforward. The names in the matrices don't always match up, such as the case here where we see on the left side, the name has this space in there. On the right side in the matrix, there's no space. And this is one of the more mild examples. There are other examples where it takes a lot more calculation and review to figure out which samples in geo match up to the matrix columns. Next slide. So we have worked on this kind of semi-automated system where we can pull all this data together and make those linkages going from the sample, the import sample to the geo sample, which is what we already get from import, but then extending that to go from the geo sample to the series, from the series to the matrix file, and from the matrix file to the specific column in that file. And we can build tables like this down here, where we have one record per link. And we can see, for instance, this experiment sample maps to this geo, accession, series, and file. And we can find its data in column 2 of that file. And that column has the name as printed here. And then we can use that data to present it to the user in this browser interface, which is a work in progress. We don't have this yet publicly available, but this is what we think it will look like, where you can go ahead and put in a experiment sample ID, and you'll get a page that looks like this on the left, where you'll see all of the different expression data that's linked to a specific sample. And so we have, in this case, there are 2 rows. There are 2 expression data sets. We have a raw count and a TPM count data set. And they link to 2 different files. And you can download the data as CSV, and we'll provide other download options as well. And that's all driven by this expression browser API, where you can, again, just put in an experiment sample ID or a geo ID, and get this type of data back that will list the different expression data sets that are associated with that sample. And this slide just kind of shows a little bit more of the functionality that we're building into this interface. So by default, you get one sample back as with a JSON response, but you can also add query parameters onto the end and say, you want to get a TSV back, or you want to get multiple samples back. Or instead of getting the entire 20, 30,000 list of genes, you can just filter by the specific genes of interest and get that returned. And so this is just something that we're building out at the moment. And it's going to enable us to use the same API on the front end to drive other analysis interfaces where we can actually get to the individual gene level expression data. And now I'm going to transition to talk about the IT infrastructure, and I just have a single slide that kind of goes over the different systems that we have in place that kind of supports all of the sites and interfaces that you've seen so far in this webinar. And so I'll start on the left-hand side. All of the boxes that you see here with the dotted borders are a set of servers that provide certain functionality. And you'll notice at the top of a lot of these boxes, you'll see things like D, S, and P, just meaning that we have a development server, a staging server, and a production server. And we generally have those three types of servers for each of anything that's user-facing. Speaking of things that are user-facing, everything that's user-facing is going to be in orange, and everything that's blue is a supporting service. So starting on the left, we have our central file server, and that's just an NFS server that is linked up to just about all of the other server sets that we have, and it hosts things like the import TSV files that we pull down regularly, additional curated data, the expression data, and so on and so forth. And we have that so that each of the servers that are linked to them have access and have this kind of common file system. Then we have our curation server, which has a SQLite database that drives the curation app interface, where our curators can interact with the data and add some additional information. The curation database gets built into this Knowledgebase database, which is a Postgres database. And we are going to put in front of that a PostgreST API interface. That's not available right now, but it will be soon. The Knowledgebase database is then transformed into a version that makes its way onto the portal server. And that's, again, another Postgres database that supports the analysis web app that Brendan demonstrated, as well as the content management system. And then I also mentioned on the last few slides, we have this expression database that's currently SQLite with a Flask API and web app. This is not currently publicly available, but also we expect to make that public soon. And then we have a forum, which we use this discourse system, which is an off-the-shelf discussion board system that we use to host the forum and our private portal. And of course, we have a clone of ImmPort that is a MySQL server. That's just simply an export of ImmPort in MySQL form. And then we also still have around the legacy portal. So this was the previous incarnation of ImmuneSpace that was transferred over to us from Fred Hutch. And that's a lab key instance that has a Postgres database that drives it. And I believe that's my last slide. And I think I'll turn it back over to Bjorn.
Thanks, Jay. Yeah, so I'm wrapping this up. As Sanchita put in the chat, please, if you have any questions, put them in the Q&A. That allows us to triage them to the best person to answer them. So to summarize some of the key takeaways here, we believe, successfully built on top of ImmPort, the ImmuneSpace interface, to provide some additional features that are still driven by the underlying ImmPort infrastructure. And one of the things is ImmPort, and for a very good reason, is not replicating what Geo does and have the whole sequencing data. So, there's linkage out there from ImmPort data into Geo SRA, and we want to allow people to tie these more directly together because that linkage is not strictly enforced. Can be a bit challenging. So that's what Jay was just showing you. We have done what I find the most exciting thing, and I've not seen anywhere else, is to actually capture, to connect the samples in the study by the way they are collected and analyzed through a study timeline, capturing things like what samples are taken pre and post vaccination, how different arms in a study are differentiated, etc. And that's what you see here on the right. And we've actually been very happy with how that has been generalizing across hundreds to thousands of studies that we've captured so far. So this seems to be quite robust and could be used more broadly. The other thing that people have been looking for a long time is signatures. Signatures here in the sense of, you have gene expression data from a certain study, but then in the whole study at the end, they might be saying, and the key takeaway here is that we have an upregulation of type 1 interferon in monocytes. So, and that is the key finding. And that is kind of like buried within the raw data. So, like kind of distinguishing the raw data and the signatures. And then again, like number 4, there have been data standards implemented within the HIPC community that are more strict, because also we have a more limited scope, that we have been enforcing kind of on top of the ImmPort standards. And actually, we've been having a great interchange with that. Lots of the standards that were originally developed for HIPCs have been taken over by ImmPort and implemented them more general if they are actually useful for the broader community. So with that, next slide, please. I think we are now just thanking people. I do want to point out Steve Kleinstein, who's the co-PI with me, and you heard from a number of people on this slide, but there's lots more people involved here. So this is definitely a team effort spread across multiple institutions. This is just another non-picture way, also pointing out obviously support from NIH. Without this, which wouldn't be possible. Within HIPC, there are subcommittees that are really helping. The steering committee has been crucial in guiding our work. And I think we have one last slide. Take a picture of this if you want, or just memorize the URLs. And I believe there's even a last thing which says, like, we do actually have a discussion forum. If you have more intricate things that you want to ask, we would be more than happy for you to enter that. And I don't know, do we do the questions next, Sanchita, or do you want to finish us up first?
Yeah, we can go ahead and take the questions.
Okay, so far, please put them in the Q&A. If you
Right.
have any more questions, otherwise, I think the first one, I believe Brandon was going to answer that.
Sure. Yeah. So we currently don't have a.
So can you read the question? Nobody else can
Oh,
see
yeah,
it.
sorry.
You have to say who asked it and what it was, and then you can answer, please. Sorry.
Yes, of course. Sorry if I'm pronouncing your name wrong. Faris Naji says what API is available to access what we are showing programmatically. Currently, there's no documented API. We're working on providing better documentation for it. It is going to be a restless API, so there's no additional authorization or requirements to use it, but it's in the works for the next releases.
Thanks.
And yeah, so there's another question from Sebastian asking, says, you said that the expression database is currently in SQLite. Does that mean that there's a plan to change that? Yes. So since it's going to be driving the API, and we expect the API to be relatively heavily used, especially by the front end, we'll run into problems with simultaneous usage if we stick to SQLite. So we'll probably move it over to Postgres.
So there's the next question from Christina asking, would most of the data available be bulk RNA-seq data? So yes, Christina, this has been our primary focus for now. We do absolutely want to have single cell data in it. That has been an extra challenge, and we kind of want to get the bulk RNA-seq data in place first, but specifically in the HIPC Consortium, actually the vast majority of data coming in is now single cell. So this is super high priority. But as you can imagine, I mean, it's already challenging to represent bulk RNA-seq data in kind of like a web form that is very useful, and the single cell data is going to be even more challenging.
And just to add on to that, we're also going back in time, and we'll be adding microarray expression data as well, because there is a lot of microarray data linked to those studies.
So I believe that's the questions that we've had. Sanchita, you want to take it from here?
Any other questions? All right, so thanks to the entire ImmuneSpace team for your excellent presentation today. Thanks, Bjorn. We truly appreciate the time, effort, and the experiences you have shared with us, and what a nice way to disseminate the ImmPort data and turn it into knowledge. So I do have, if there are no other questions, maybe I'll take a minute to for some announcements from the ImmPort side. Let me share. I do have, okay. Let me go back. All right. There you go. Oh, went away. Sorry about that. I'm hoping people can see the slide now.
Yes.
So a quick announcement, information that all these webinar recordings, slides, and additional materials are available for download and viewing on the ImmPort documentation website. Here is the QR code and the link available. So those who couldn't attend or would like others to view, we will make these recordings available in a couple of days. And if you want to go and check out the other webinar recordings, please feel free. This is the website. And stay connected with ImmPort for upcoming webinars and explore new data released through our mailing list. Here is the office hours. If you have any questions about ImmPort data analysis, we host first Thursday of every month from 2 to 3 p.m. Eastern Time, office hours. And then we do have information coming out through the newsletters, where you will get to know about new data, new features, and ImmPort events. So feel free to register to the newsletter. And we have a social presence as well, so all the links provided here. And with that, if you have any further questions, please reach out to us through the ImmPort help desk. And I also would like to thank everyone for joining today for the webinar. We appreciate your time and interest in learning more about ImmPort and our commitment to supporting immunology research. Once again, I would like to thank the ImmuneSpace team. Also, I would like to extend my thanks to the ImmPort support team for all your help and coordination. So with that, I think we will be able to give you 10 minutes back. Meanwhile, let me check if there are any other questions for ImmuneSpace or for ImmPort.
Yes, I saw a question here from Reed and is part of the goal to identify novel signatures through ImmuneSpace after data deposition. Will be interested to see if this platform allows additional insights. So 100%, that is the driving goal. The whole reason for funding kind of the HIPC consortium as a consortium was to then cross-compare between different perturbations, so different kinds of infections, different kinds of diseases and interventions. How do the signatures look like? Can we identify more common patterns across these things, across the original studies that are that they're showing up repeatedly? And there have been a number of there was a series of Nature papers, Nature Immunology, etc. papers that came out, I think like 2 years ago from the last cycle. We hope to do the same, that we're doing like these kind of like meta-analysis across the HIPC data that was then done essentially through like the precursor of the current ImmuneSpace platform, which at that point was pretty much accessible only to HIPC consortium members involved in the analysis. So now we're trying to make these all in the open so that others can directly reproduce and do their own analysis and do more analysis that we didn't think of. So, yeah.
Thank you. All right. So, okay, so we are coming to the close and we'll see you in the next webinar. Thank you. Thanks, everyone. All right, so I'm going to end the seminar. Thanks, team. It was amazing. A lot of work has been done. It's great to see that. Thank you.
Thank you so much for having us. Appreciate it.
Thank you.
Thanks, everyone.
Thank you.
Bye.